Hello students, today I am going to discuss about the second part of the bacterial recombination that is known as transduction. The transduction is basically the transfer of DNA via the bacteriophage. When the bacteriophage is used as a vector of DNA transfer from one bacterium to another bacterium, that is known as the transduction. Basically, this is the type of horizontal gene transfer. The transduction mechanism was first discovered in 1952 by Norton Zinder and Joshua Lederberg. And due to this experiment, along with the discovery of conjugation, Lederberg was awarded in Nobel with the Nobel Prize in the year 1958. Basically, this is the experiment what they have executed. This YouTube have to open and the bacteria known as the Salmonella species, the Salmonella typhi of the bacteria. This bacteria was used in this experiment for the discovery of transduction. Basically, they were the experiment was going on for the transformation, uh, but they also cultured here the phage P22, which is specialized phage for the Salmonella strain of bacteria, Salmonella genus of the bacteria. In the middle of this arm there was a glass filter, and the strains used two stain were used in this experiment. One is La22 and another one is La2. These are separated by this glass filter, and no bacteria cell can pass through this filter. But when they plate on the minimal media and incubate, they found that no growth no prototropes were found, but here the growth of prototropes can be found. So the conclusion was that as the patch P22 was small enough to pass through the filter, and it infect the La22 and incorporated in La2 DNA, and thus this made the La2 phase auxotrop, and it also convert the La22 phase in as the prototropes, and so the prototropes can grow in the minimal media on the plate. Basically, there are two types of bacteriophages. Those are infected the bacteria. The phage that infects the bacteria is known as the bacteriophage. There are two types. One is lytic or virulent, and another one is lysogenic or temperate phage. The virulent phages are multiplied within the host cell and lyse the cell and release progeny phage. As for example, the T4, T7 are the examples of lytic phage, and the lysogenic phase has the typical characteristics that they transfer their genetic material inside the bacterium, and after transfer the genetic material of virus can incorporated into the genome of bacteria, and this is known as the lysogeny. When the phage DNA is inserted into the uh, host bacteria host genome, and it forms the quiescent state, it is known as the prophage. This type of bacteria carrying the lysogenic phage DNA, when they faces some scarcity of nutrient or any type of adverse condition, they they resume the lytic cycle, and the phage is released in the nature. So basically, this is the lytic and lysogenic cycle. The comparison is shown here. This attack of the virus was done here, and after that, these bacteria may this phage may inserted into the lytic cycle and produce so many fudge particles, and those fudge particles degrade the bacterial genome. And after that, using the host cellular machinery, they produce the coat proteins and head and tails of the fudge. And after assembly of these fudge particles, they released from the bacteria by lysis of the bacterium host. And either in the second one pathway is that is the fudge genetic material can incorporated into the host, and it undergoes the bacterial life cycle. And when the adverse condition comes, they go to the lytic cycle to produce numerous fudge particles. Basically, the transduction is the gene transferred from a donor to a recipient by a way of the bacteriophage. Here, the bacteriophage is used as the vector, and this is not like the transformation what the naked DNA is transferred into the bacteria from one bacteria to another bacterium. And so there are three types of transduction. One is generalized transduction, second one is specialized, and third one that is less understood till now is the lateral transduction. It is only described in Staphylococcus aureus. The generalized transduction is can follow the lytic cycle of the fudge propagation, and specialized transduction follows generally the lysogenic cycle. In case of lateral transduction, this is less understood, but this is the only transduction method by which the more genes can be transferred into in a higher frequency than generalized and specialized transduction. So come to the point that the structure of bacteriophage, we have to know, we have to understand the little bit for the more knowledge. The what is the structure of bacteriophage? Bacteriophage is the typical phage that infects the bacteria only, and it has one head and another tail portion. And at the tail portion, we can see there are some spikes and the base containing some pricking molecules or some short piercing molecules of the structures are there. With the help of this piercing structure, the bacteriophage produces holes on the bacterium cell wall, and the genetic material is located in the head. And after it sits on the after sitting on the bacterium cell wall, the head squeezed its genetic material through the tail portion, and through the hole created by this base plate, the genetic material is transferred into the bacterium cytoplasm. So this is the mechanism discussed in a little bit detail. It sits on the bacterium cell wall due to the property of adsorption, where the tail fibers are acts as the helping instrument or helping organ for the adsorption process. And the base plate is attached with the bacterium cell wall, and nucleic acid is injected by the squeezing method, and DNA is uptaken by the bacterium cell wall. So this is the electron micrograph of T4 fudge. The fudge DNA is carried by two types of fudge known as T4 and known as virulent and temperate fudge. I have discussed it previously. The examples are virulent fudge is T4, T7. The T series fudge are the virulent one, and the temperate example is the lambda. This is the special case of bacteriophage, where we can observe both circular and linear chromosome in linear genetic material in case of this temperate phage. So come to the point of generalized transduction. It is the process by which any bacterial DNA may be transferred to another bacterium by a bacteriophage. It is a rare event, and very small percent of phage particle happen to carry a donor bacterium's DNA on the order of one phage in 10,000. The essence is this is the packaging of bacterial DNA into the viral envelope. This may occur by two main ways: recombination and headful packaging. What happens in this process? When the bacteria is getting genetic material from a bacteriophage, the there is by chance one bacterial DNA can be packaged in the head of the virus particle, and when this virus carrying the bacteria genetic material is when it attacks on another bacteria, the genetic material transferred into the new recipient bacterium. When some homology is found between these two genetic materials, the bacterium, the virus DNA, which is actually the bacterial DNA, get inserted into the genome of bacteria, and new recombination uh, was happened. New recombination is happened, and this is the main essence of generalized transduction. By chance it occurs by chance, but when it occurs, the new recipient bacteria gains the property of previous donor bacteria. Now come to the second part, which is the specialized transduction. In this process, when a phage DNA, that is the lysogenic phage or temperate phage, infects a bacterium, it inserts its genetic material into the host bacterium cytoplasm, and the genetic material of virus is getting inserted into the host genome. And after that, this host genome is known as prophage. The embedded the genetic material embedded in the host genome is known as the prophage. If this prophage is undergone the any type of nutrient deficiency or any type of adverse natural condition, this bacterium produces the lytic cycle of the virus regeneration, and here accidentally one virus genome is packaged with the recombined genome of from bacteria, and this virus particle, when infects another bacterium, the genetic material, the modified genetic material, is inserted into the new host genome, and thus the recombination occurs. So basically this is the mechanism of specialized transduction, and the process of transduction is nothing more. And let's take some example of bacteria those recombines using the transduction methods. Those are Disulfo Vibrio, Escherichia or E. coli, Pseudomonas genus, Rhodococcus, Rhodobacter, Salmonella, Staphylococcus, and Xanthobacter, as well as Methanobacterium and some Thermoautotropicum are there. Those bacteria reproduce or through via the recombine via the transduction method. Thank you.